So I've updated some settings, so I hope this works better today than it had yesterday. But you know, we'll find out. I deleted the last video. I deleted Monday's video because of our streaming problem. So it's no longer there. But nonetheless, I'm not really saying anything massively unknown, right? So just going to Ensemble and downloading sequences. So we're going to keep playing with that, and furthermore, we're going to slowly introduce analyses we can perform on sequence data. Now, I went and grabbed, previously I grabbed a human PWL1 and a bonobo PWL1, but then I looked at those sequences and they're too similar to really do anything. So one, recognize like these, us and bonobos are separated by about 5 million years. And this protein sequence, not only the protein sequence, but the DNA sequence is effectively identical. So it's a pretty conserved gene. Nonetheless, so I can't compare it because it is identical. So I'm going to grab a different sequence, or a PWL1 from a different animal, to go through some analyses, or at least some simple concepts related to DNA analyses. So I'm going to get a dog sequence. So I'm just gonna click Larysotheria because I know it's a Larysotherian, and so I don't have to scroll through this whole thing. So I'm just gonna get the first dog, and there's more than one dog on Ensemble. Like a dingo is available, a Basenji is available, a Great Dane's available, but the OG dog, it doesn't say, but it's a boxer. The OG dog is a boxer. So I'm just going to go get this PWL1. Again, export that sequence. I want this coding sequence, not the cDNA. And I'm just going to get that real quick. And I'm going to paste it into my working text file. Okay, so here's my working text file. I already have my human PWL1. I got rid of the bonobo PWL1. Again, it's not very helpful. So I pasted in my dog, and of course I'm going to change my my header information so that I have the accession number from Ensemble. I know that this is a dog, and I know it's a PWL1. Okay, so I'm going to leave it at that for now. We'll gather some other sequences later, but just to go over some basic concepts about DNA analyses, I'm just going to use these two sequences first. And I'm also going to introduce Mega as well. So first of all, I'm going to save this file. I'm going to save this in the same kind of test directory that I've been messing with for our R scripts, just so I know where it's all at. I may change this test to like bioinformatics, because this seems to be where I'm saving all of my stuff. Anyways, I'm just going to name this something descriptive, so whenever I look at the file, I know what it means without having to open it. So I might do like mam pwl1 test.fast. That may be a little bit tricky to see on my window, but I'm kind of circling back to the importance of naming extensions, or having appropriate extensions on file names. So I use .fast. That lets me know that this is a Fasta file. So you will traditionally see Fasta files have an extension of .fast or .fasta. So I'm just going to save it there and leave it alone for now. So I guess I don't know if this is really if important is the right word, but I guess just to, I don't know, remember what word, properties of DNA. Just reminding ourselves about some properties of DNA. So if I'm reminding ourselves about DNA, really even transcription and translation, right? By this point, we should have all heard at least transcription and translation and processes, those processes at least twice by this point. So I'm not going to go over a whole lot of detail, but I am going to remind ourselves about properties of DNA. So one, it is double-stranded, and it's an anti-parallel orientation. And we determine orientation based off of a 5' to 3' orientation. All right, so that's going in forwards and reverse. So this bottom strand is going to be in the opposite orientation, so it'll be 3', and this is 5'. Right, and so our bases are complementary. So we may have ATG, CAA, TAC, GAT, whatever. And the strand is going to exhibit complementarity. Oh, I should have drawn this wrong. Ignore what I'm doing. I'm going to change this so I'm happy. Yeah. Okay. And so there's going to be complementarity. So this will be T, TAC, GTT, ATC, CTA. Right? I'll review stuff. And so recognize, like, double-stranded DNA is effectively redundant. Right? So it doesn't matter if I have, if I use this top sequence or the bottom sequence, I can recreate both. Right? So if I just have like my bottom ATG sequence, if this is all that I had, I could still replicate the complementary sequence by doing a reverse complementarity. Like I can switch the orientation and change my bases. Reverse complement, that's the word I'm looking for. Reverse complement the sequence. So you'll see in databases that we only get one strand because having two strands is redundant. And also, we should recognize that whenever we ask for a gene sequence, we get the coding sequence. So, right, remember, like in transcription and translate, or in transcription, we do have these two strands. We have a coding strand and the template. And the template will be transcribed. And the mRNA is effectively equal to the coding sequence. Right, so basically whenever we transcribe the template strand, we're ultimately going to get this sequence as an mRNA. Right, so our mRNA will be like AUG, CAA, UAC, etc. Right, the only difference between the mRNA and the DNA and the coding sequence is that we replace our T's with U's. But also an mRNA sequence is kind of redundant. Right, if we have the coding sequence, we really don't have to make the mRNA to know our protein. We can directly translate the coding sequence to an amino acid, right? So we can grab these codons. If we've forgotten that word, we can grab these codons and figure out what they translate to, right? What amino acids they translate to. I don't know. The only one that I know that I could get out of that is the first one's going to be methionine. The others I'd have to look at a translation table. But if you get good enough at this, you don't have to even look at a translation table. You just make a program to do it for you. So there's no reason to memorize all the codons and how they're translated. Anyway, so whenever we download these sequences, we're getting the coding sequence. Again, because we can just work directly from them. If we wanted to get the reverse complement, we could. Nonetheless, right? So all gene sequences are going to just inherently be ATG to stop, right? So now I'm going to kind of take a small, brief tour of Mega and how we can upload these sequences to Mega. So all of y'all should have this by now. If not, today. Because we will keep using this program more, certainly for the first couple of weeks. Because what we're going to do is we're going to mess around with DNA sequences, but we're going to use Mega because we can look at our manipulations. We can look at the things that we're doing. Whenever we switch over to the Linux terminal, we won't be able to see what we're doing. It's all going to happen behind a screen, and we won't be able to observe what kind of changes we're making. So Mega is nice first as an introduction to these types of analyses. All right, so if I want to load in this FASTA file, probably the most the canonical way to do this is, we just go to File and Open File. So Open File Session, and then I'm going to go find that FASTA file. I know that's on my desktop. It's in test 2, and it's this MAM PWE test. And so the nice thing about Mega will inherently read a FASTA file. So as long as your file is named, it has that .fast extension, you really can just double click on it and Mega will open it for you. So that's kind of a nice property about it. But nonetheless, canonical way. The best, probably the best way to do this. So just open that file up, and it's going to ask you, do you want to align this sequence or do we want to analyze this sequence? Well, we're not ready to analyze this sequence yet, or these sequences yet. So I'm going to do align. Because what we'll see, so here's our dog and our human peewees, right? So we know where those came from. We know which is which. All right, so this is why we define our header. We try to make our header fairly descriptive. So whenever I look at that header, whenever I look at these sequences, I know which sequence is which. Anyways, so whenever we're looking at this alignment view, any bases that are identical will have this asterisk above it. But if we have a mismatch, so like in this first AT, I might be able to make the font a little bigger. Let's see. Yeah, let me try 12, just so we can see it a little bit better. Right, so we have this CNA mismatch, so we don't get an asterisk over it. We get another CA asterisk that we don't have an asterisk over. Okay, and so if we look at this sequence, it's reasonably identical, reasonably similar, I should say. But at some point, we'll see that similarity kind of drop off. Okay, so around this point, we start to see some similarities drop off. So around this area, about midway through, these sequences stop being so similar to each other. And it's not that these sequences are so different. It's because these sequences are not aligned. So, which means Like, we have to estimate base homology. So like, let's see if I can draw this out. Let's see how well this goes. So if I draw out this, like a short version thereof. So let's say I have ATG, CAA, GAT, and then so that's sequence number 1, and sequence number 2 will be reasonably similar. But let's say ATG. Okay, so this sequence is shorter. And if we recognize like that codon is identical, this codon is identical, but these codons are not identical. But these two codons are identical, and that one's identical. And so, but if we try to do any analyses on this stuff, we're going to compare these bases. So we'll have, I'll do this in another color. So let me do red. So that base will be compared, that base will be compared. All of these bases will be compared to each other, but because of this extra codon, we're not doing a homologous comparison of these sequences, right? So this codon is not identical to that codon, so but we're going to compare them anyways if we don't align them, and then we'll have nothing at the end for TAA to be compared to. Okay, so ultimately, if we don't align these sequences, if we don't account for this weirdness, we're going to get an overestimation of sequence differences. So what we have to do is we have to align it. And it's effectively we insert gaps where non-homologous bases are. Again, the whole idea is to identify and estimate homologous base pairs. So this would be unaligned. But if I align this, what we'll see is ATG, CAA. Okay, and so whenever we align this, we account for this gapped codon. Right, we have this one codon that doesn't seem to be in place. It doesn't seem to fit in this sequence very well. We could, from this point of view, we could either assume that this codon was inserted in sequence 1, or the codon was deleted in sequence 2. We really can't tell. And so now we can actually do accurate comparisons of these bases, right? So now we can, all of these bases will make sense with each other, right? Now we have an accurate comparison of these two sequences. We're going to ignore this gap, but nonetheless, when we take this into consideration, now our sequence similarity will be much higher than if we calculated it from raw sequences, right? Because these bases are not homologous to each other. Now I'm not going to go into the math and the programming behind how we do alignments. Besides that, it's called a dynamic programming table, or dynamic programming. And I may send you a video about that kind of algorithm, but it's tricky for me to explain. And it takes a while to do so. So I don't want to use class time to poorly explain dynamic programming. But nonetheless, Mega has a built-in aligner. And so in evolutionary biology, in bioinformatics, there are at least a dozen different aligners. The first one was Clustel, Clustel W, and so that's still implemented in Mega. Right, so Mega, well, under the alignment tab in this alignment view, we have aligned by Clustel, Clustel W, aligned by Clustel W codons, aligned by muscle, or aligned by muscle codons. And so these two aligners are inherently built into Mega. Right? Mega didn't make these aligners. These aligners were already made. The writers of Mega just incorporated those tools into this software so we can do stuff easily. Now, whenever we align proteins, whenever we align protein sequences, anytime we align coding sequences, whoops, hold on. Whoops. Coding sequences must be codon aligned. Right, so this is important. Anytime we study the evolution of protein coding sequences, we have to do a codon alignment. And we might see a reason for doing so. I'll try it both ways. I'll try Mega both ways, just doing a regular alignment or a nucleotide alignment and then doing a codon alignment to show these differences. Oh, okay. There we go. All right, so we have all this gap sequence at the end. So we know that this dog sequence is a little bit shorter than the human sequence. Okay, so I'm going to codon align this. So I can either go to alignment and align by muscle codons, or I can just click my muscle button. Right, it looks like a bicep. So I can just click that button, and it will give me align by DNA or align by codon. So these two buttons are probably by far the most ones I use. Is this W button or this muscle button? But even then, I don't use a Clustel aligner anymore. It's not that it's bad, it's just that it's kind of primitive. And honestly, I don't even use muscle aligners very much either, for kind of the same reason. But nonetheless, this is what's built into Mega, and I do think muscle is a better aligner than Clustel, and I hope the internet doesn't yell at me for that. But nonetheless, I'm going to align by codons. Yes, I want to select the entire thing, and so it's going to give me this window of options. Kind of like a rule for me, I almost always use default values. And the reason I use default values is because anytime you mess with these parameters, your alignment will change. And if you don't know what you're doing, you can make your alignment much worse by touching these parameters. And so the default is kind of like the best case scenario for everything, although it's not going to be great for everything. Nonetheless, it's just kind of, especially as we go through this class, unless I tell you otherwise, just use default values, because it probably is going to work as good as we need it to. Anyways, so I'm just gonna, don't care. Yes, I'll remove my gaps, and it's going to take like 2 seconds to align. And so it's going to insert those gaps where the alignment doesn't match anymore, right? So if I scroll through this alignment, I should have a couple of gaps, right? So I have a couple of gaps here, and quite honestly, I would put $1 on that this isn't really a gap. It's just that whenever we try to annotate a 13 or 14 exon gene over a spell of 100,000 bases, one of those exon annotations is going to be wrong, at least. Or it's going to be only partially right. So regardless, we have a very clean alignment for the rest of this, except for this little bit of gappy stuff. Now, if we take note, these gaps are in replicates of 3 for codon length, right? And even over here, so this will be these gaps will be a multiple of 3, 3, 6, 9, and 12 to reflect 4 codons. Okay, and then everything looks nice, and our stops are in the right place. Yay. Right, so our sequence is the same, pretty much the same throughout, except for this little gapped, this slight gapped region. All right, so I'm going to redo this, but just as a nucleotide alignment. Let's see what happens. Okay, let's find that gappy region. Let's see what happened with my gaps. Uh, 1, 2, 3, 4, 5, 6. 1, 2, 3. It still did the same thing. So it didn't do a codon alignment, but this did not work as I was intended. So if we do a nucleotide alignment, so what we do whenever we do a codon alignment, behind the scenes, what's actually happening is we're going to take this coding sequence, we're going to take these coding sequences, we're going to translate them to amino acids, and then align the amino acids. And then return the DNA sequence. And we do a codon alignment for protein coding genes because it's the protein that faces selection. It's not really the DNA sequence, it's the protein that functions. It's the protein that will experience selection. And so it's really the protein will evolve, and the DNA sequence will always stay as a codon sequence, as a codon. Right? Because if we try to align this as if we tried to do the same thing, if we took these 3 sequences, this is going to be, I don't know if I can do this. If we did the same thing and did a codon, a DNA alignment, theoretically what would happen? Right? This could be a hypothetical alignment of our two hypothetical sequences, where ultimately we'll get a mismatch. Our gaps won't be 3 bases long. Our gaps will only be a single base. And for a codon, for a protein coding sequence, that makes no sense to have a gap that's a single base long, because an amino acid in a codon is 3 bases long. And so we have to account for these amino acids. We have to account for codon position when we do alignments of coding DNA. Now, if we're just picking like intron sequences or UTRs or TEs or something that's not coding, yeah, we do an alignment. We do a regular DNA alignment. But if it's coding, it's the codons that evolve. It's the codons that change, not the raw DNA sequence. Anyways, back to this, back to our mega stuff. So I'm gonna get rid of this just in case it's weird. Yeah, let's see what happens. Okay, nope, stupid. Okay. Okay. Now we have our codon alignment again. And the nice thing about Mega again is it has a built-in translator. So I can just go to translate proteins. It's going to ask me if this is a standard genetic code. It is, because all nuclear DNA through all domains of life have used this standard translation table. So I translate it. Now we see the actual amino acid sequence. And so whenever we see the amino acid sequence, we can see those gaps are really, they're there just for the amino acid, right? So they're gapping the amino acid, they're gapping the codons and the amino acids to produce our alignment. Normally, under a better situation, I would get two sequences that would align okay, but we'd have these gaps of single bases or two bases, and it's going to disrupt our rating frame, and it'll look awful whenever we translate to DNA or translate to protein details. Anyways, so honestly, so if we kind of take a look at the sequence, like the DNA sequence is kind of variable, right? We see some asterisks and not so many asterisks. But whenever we look at the protein, when we look at the amino acid sequence, oh my gosh. We see a lot more asterisks, right? Because what we're observing, what most of those changes are going to be silent mutations or silent changes in the third codon position. All right, so one simple analysis we'll do, I'll come back to that probably on Friday, but one simple analysis we'll do is that if we group again, we're reminding ourselves about some simple DNA stuff. So if we're reminding ourselves of some DNA stuff, remember our nucleotides get broken down into, we have two types of nucleotides. The purines, and so these babies are pure, they say GA, this is guanine and adenine. And the pyrimidines, oh, I'm not going to be able to spell this, holy crap. Pyrimidines is going to be the CUT. Pyramids are sharp and they cut. So in DNA, that's cytosine and thymine. And given this kind of information, we'll see two types of mutations, or two types of substitutions. We'll see the transitions that are like a G will be substituted for an A, or C will be substituted for a T, right? These are transitions. And alternatively, the transversions This is where we're having, where like a G will, we have a purine to a pyrimidine or vice versa. Really, I mean, right? So a transversion is when we have a pyrimidine to a purine or purine to a pyrimidine, right? Kind of transversion. And when we look at DNA sequences, these will be way more common because this more often than not, especially in coding sequences, there are less consequences for a transition than there is for a transversion. So we can, with Mega, we can estimate a transition and a transversion rate, or we can at least count the number of transitions and transversions between these two sequences. So if I do a quick, I can't analyze these sequences in the alignment form. I can save this alignment if I so wanted to. I would just go to data and then export alignment as a Fasta format, but I don't necessarily want to do this. I want to keep this data in Mega. But I can go to phylogenetic analysis. I can just go to data phylogenetic analysis, and it will just inherently load my sequences into Mega. So like this is my alignment file. That's going to be my alignment file. And this is going to be my data file that Mega can work around with. Right, so I have my alignment and my data. So now that my data is actually present in Mega, I can now kind of play with around these other buttons. So we can look at sequence diversity, we can compare sequence distances or how different they are from each other, and we can reconstruct phylogenies, etc. We only have two sequences, so we really can't do that. But the first little thing we can do is estimate how many transitions are present in this alignment. So I can go to distance and do a compute pairwise distance, and that's going to give me a chance to count the number of differences, right? It seems like OneNote's kind of a problem, or is causing me more problems than Mega is, so I'm going to leave OneNote alone. I may stop drawing, or at least I'm not going to draw on OneNote anyway. It may be too much for all of this to handle. I seem to be doing better whenever Mega's running. Anyway, so we have all these options. Now this is stuff that we can mess around with. So we are working with substitutions. This model method, a lot of different ways to calculate sequence differences. A lot of different ways. But for now, I'm just going to count the number of differences, right? I'm just going to do a raw count. And specifically, the substitutions that I want to look for, I'm just going to count the number of transitions only. So I'm going to run that, and now it's going to give me this pairwise matrix, and I'm going to have, it's going to tell me there are 197 transitions in this alignment. Yeah. And the nice thing about this little spreadsheet, about this little thing, is that it will tell you what you ran. So these two sequences were compared. I did a number of differences and transitional substitutions only. Okay, so I'm going to leave that there. So that will stay in my little mega window. I'm going to do it again. Do a pairwise distance, okay, and I'm going to count transversions only. Let that run, and it's going to tell me I only have 76 transversions in this alignment. Right, so this is like the first very, very simple analysis, and it validates what we've already been told. Like transitions are way more common than transversions, because transitions, one, they're easier to happen. Right, substituting a G for an A isn't nearly as is simpler than substituting a G for a T. So transitions are always going to be more common in sequence differences than the transversions, right? Validated, right? You've heard that before, just validate it. And that's going to be true for effectively every sequence that we mess with. Any pairwise comparison, the transversions will always be less than the transitions. So I'm going to leave those two there. I'm just going to do one kind of quick little test and just see what happens. Just see what happens. Yeah, that's not good. Okay. All right. First thing we've ever done playing around with Mega, doing some sequence measurements, right? Just counting the number of differences between two sequences and what kind of differences those are. Neat. But I'm gonna leave it there. So I guess I'll go ahead and save this alignment as a FASTA just so we do it. Export alignment, FASTA format, and then I'm going to change this. Oh, different directory. So I'm going to do like mam pwheel1 align.fast. So I know in that file that it's an alignment and it's in a FASTA format, right? Again, just trying to describe the file as well as I can so I know what's in it without having to look at it. All right. I know it was a little laggy today, but I think it was better than Monday. And we might have found a pattern with when it gets a little laggy. So I'm going to leave OneNote alone for a while. If I have to draw, I may use PowerPoint or something and see how well that works. All right. Until Friday, we may collect some more sequences and do some more analyses. All going to be PWL1s, all going to be mammals. And we'll see what happens.